Hey Zoology, this is our video on Phylum Porifera. So Unit 2, Phylum Porifera, these are our sponges. In Unit 2, we're going to be covering sponges and all of the jellies, but in this particular section, we're just focused on those sponges. So a couple of things. We watched this the video to this link in class. Couple things to remember, this is not in your notes, but sponges are the simplest multicellular animals. So they are kind of the original animals. They live on the bottom and they attach themselves to things and then they never move. So they're sessile. So that might seem a little weird because you're like, wait a minute, aren't animals supposed to be able to have movement? Well, yeah, at some stage in their life. Porifera larvae, or sponge larvae, basically the kind of embryonic baby form of the organism, is able to move around. And so that is where it kind of gets that complex movement. So we see there's a lot of variety in sponges. We can see that there's some large barrel sponges, some huge barrel sponges down here. Look at them compared to the divers in the background. And then we have some tube sponges, some encrusting sponges, and some other kind of just squatty type sponges. So there's a lot of different types and they are ancient. They've been around for millions of years. So the question comes up, okay, they just look like rocks or plants. Are they really animals? Well, yeah, but they are early animals. So like animals, what they do have in common with animals is they are multicellular, they are heterotrophic, they do reproduce sexually. But unlike other animals, they do have some kind of differences. They don't have complex movement when they are adults. They're sessile as adults. Sessile means kind of attached to a surface. They don't have an intestinal tract, so their blastula actually does not undergo gastrulation. So we said there are those animal architecture things, those 6, 1 through 6 Roman numerals. Those are kind of the things that help separate animals into their different phyla. And so sponges were separated because they don't undergo gastrulation. So as a result, they don't have any dermal layers, and they are only organized on the cellular level. They have no tissues. So general characteristics of sponges, they are immobile, they're sessile, they don't move. They're filter feeders, so they use collar cells to actually, the flagella in the collar cell wiggles and kind of pulls the water through the sponge, and then the food gets trapped in the collar around the outside of that choanocyte or the collar cell. And so they just are gathering little particles of food from the water around them. The term porifera means pore-bearing or many pores. So they do have a lot of openings on the outside. We'll see that they're kind of organized differently depending on the type of sponge it is. But they have those pores on the outside and water is pulled into the animal and then it is passed through that collar. And as it passes through the collar, it sticks to it. There's a mucus on the collar of the collar cell or of the choanocyte. And so the food gets stuck in that mucus, and that's how they eat. So that food, once it's stuck in the mucus, it passes through back to the amoeba site, also called the archaeocyte, which we'll see a couple times, but we'll talk about those. And that's kind of the actual cell that consumes and survives off of the food. So most of them are found in the ocean. Only about 150 of the 15,000 species exist in freshwater. And they have 2 major types of body openings. We have in-current pores, in meaning that's where the water is coming in. Those go into the ostia. And then the sponge usually either has one large opening for the water to flow out, or it has just larger openings for the water to flow out, and those are called osculum. So the in current are the inhale, essentially, the bringing in of the water. The ex current is the exhale or the exiting of the water. So inhale is ostia, and there is an I in ostia, so that's a good way to remember. That's ostia, that means it's coming in because there's an I. Osculum, it's going out. So here's kind of a diagram of how that looks. So the water comes in through the ostia on the sides of the sponge. This is kind of a basic body style for the sponge, you'll see. But it does that, and then as it passes through the pores, you're going to see that it'll pass by and over the collar cells, and those flagella pumping push the water through the collar. That's it's almost like a colander or a strainer. And so it will hold the food in there and the rest of the water can pass through. The food particles stay back and then they are consumed. And then the water is pumped out through the osculum. So sponges are organized around a canal system. So again, it's all about water movement. And so you have that water coming in the ostia, out the osculum. And the choanocytes, also called the collar cells, are the ones that are pumping that water. So they are actually moving their flagella, and as they're doing so, they're pumping that water through. So they communicate enough. They're not a tissue, but they communicate enough as cells to make sure that they're pumping in the same direction at the same time. And so that pumps water through, and that's how they kind of sieve out their food. And the body is supported with a endoskeleton of spongin, which is a type of collagen. Collagen is the substance in our face that allows us to do things like this. And then when we let go, it goes back to normal. So collagen is kind of that elastic fiber. We have a lot of collagen. Spongin is very similar to collagen. So it's very similar purpose for them. And then they also use spicules made of either calcium carbonate or silica, which is a type of glass. So in this picture, we see the spongin fibers. So those are kind of the wiggly fibers. And then these darker kind of sharp pieces are the spicules. So usually there's not a whole lot of organization. It's just kind of the spicules form where there's deposits of calcium carbonate or silica, and then the spongin forms and grows, and that's how we create the sponge. So it's essentially kind of a spongin skeleton with spicules that kind of hold it up like scaffolding. So it's like a, just kind of a shell, if you will, kind of a shell of spongin, and then those spicules are just giving it extra support. So there's three types of canal systems in sponges. The first is asconoid. Asconoids is the simplest type. They are usually just tube-shaped sponges. Water enters through the dermal pores, through the ostia on the outside, flows into the interior, out the osculum on the top. So that's kind of your basic, typical type of sponge. Here's a picture down here. Let me hide myself for a second. Here's a picture down here of some asconoid sponges. The second is ciconoid. So ciconoid sponges are a little bit more complex. You'll notice on the outside of a ciconoid sponge that they aren't smooth. They have like a lot of roughness to them. And the reason they look rough is because the outside of the sponge, there's essentially kind of an increase in surface area. So instead of it just being, instead of the outside of the sponge just being, so in an asconoid sponge, the outside of the sponge is literally just a series of openings where water can just flow right in. In a ciconoid sponge, it's essentially more surface area, so water has more ability. Oh, these are not arrows, but hopefully you get the idea. It's essentially the similar idea, but with more surface area so that more water has the ability to pass through into these little chambers and then into the overall center chamber of the sponge. So the, again, asconoid, the first type we looked at, are just basic holes on the outside, water flows in, pumps out the top. Ciconoid are almost the same, but they're just a little bit more of a complex version of the asconoid. So they still have that single osculum on the top. They still have that single opening for water to go out, but they have a lot more of a kind of complex exterior. The body wall is thicker and more kind of complex. The last is the leuconoid type. This is the most complex. Leuconoid sponges have a lot of different osculums. So asconoid and ciconoid sponges only have that one osculum on top, so they pump water in, it goes out the top. Leuconoid sponges are more complex. They have a lot more of an internal chambers, internal passages, and a lot more osculum. So water can pass through in a lot of different locations within a leuconoid sponge and pass out of a lot of different exits. So sponges are suspension feeders, so they pull food from the water around them. It passes through their choanocytes or through their collar cells. Those collars are covered in mucus, and so the flagella pumps. It pumps food over the collar, the food sticks to it, and then it moves it down to the cells called the amoebocytes. So those are kind of the actual energy-driving cells of the sponge. So make sure you're understanding this process. It's definitely something that you could be asked to explain in an essay question about how does a sponge get food from the water. They pump water flows into the ostia. They pump water using the flagella on their choanocytes, and that passes it through the collar, sticks to the mucus, passes to the amoebocyte, and that's how they get their food. So sponges have the ability to reproduce asexually. They also reproduce sexually, but asexually, essentially, it's a bud formation. So they are essentially taking little parts of their body and budding off to create a new sponge. Now I put here regeneration because this is also a way if a sponge has a portion of it that's eaten, it can actually regrow that area of the sponge. So it creates these things called gemmules. Gemmules are essentially amoebocytes, also called archaeocytes, but same thing, covered in spicules. So essentially, they're little pockets of cells that are ready to eat. And then as it lands on the ground or on the bottom of the water and it starts to grow, it will then develop into a full sponge. It'll develop its ostia, it'll develop its osculum, and then it will be able to start eating. So it's essentially just a little pocket of sponge ingredients, and then they fall off. In this picture over here, you can see here's a bunch of them on the outside of a freshwater sponge, and then SpongeBob budding off. You get the idea. They also can do sexual reproduction, but most sponges are hermaphroditic, meaning they have the ability to produce sperm and eggs. So a lot of times they'll just sexually reproduce with themselves, although that doesn't allow for a lot of genetic variation. So they will, if they are around other sponges, they will undergo sexual reproduction. If they're not, they'll usually just do asexual because it's easier. So the sperm are released, and then they can enter through the ostia of another sponge, and then as they pass through the body, they land on the eggs that are in that one, and then they can go from there. And then they create larvae that are ciliated, meaning they can move around. So this is an example. All of these little hairs on the outside of this are little cilia that kind of pump and wiggle, and it keeps it moving around until it finds a location it wants to land on. So here's kind of a diagram to show that sponges are capable of sexual reproduction. So again, sperm and egg come together, and they can either be happening within the same sponge or in another sponge. And then they fertilize, create their blastula. They do not undergo gastrulation, but then they create their larvae, settle, and create a new sponge. Or they can just bud off, it floats away, lands, creates a new sponge. So there's 3 different classes of sponges. The first is calcarea. And a good way to remember calcarea is that they are calcareous. They have calcium carbonate, calcium carbonate. So calcareous sponges have calcium carbonate spicules, and you can find asconoid, seconoid, and leuconoid sponges within class Calcarea, but what sets them apart is their calcium carbonate spicules. Class Hexactinellida, that's a rough one. Hexactinellida, you don't have to pronounce that one because I can't. Good thing to remember about those is that they have silica or glass-like spicules, but specifically within this class, they are 6-rayed. So they are going to be looking like this, where their jacks kind of looks like jacks, if you've ever played marbles and jacks. But that's what their spicules look like inside of them. Most of them are going to be seconoid. And then the last is the class Demaspongiae. And these also have silica sponges, but they are not 6-rayed. So hexa is the prefix meaning 6. And so the Hexactinellida will be the ones that have 6 rays, whereas Demaspongiae are not. So class Calcarea have calcium carbonate spicules, and then the other 2 classes have silica spicules. The one that has silica spicules that are 6-rayed are going to be the ones that are found in the class starting with hexa. The others are in Demaspongiae. These are most of the sponges, but here's kind of a diagram that shows you what we mean about the different types of spicules. So class Calcarea has calcareous spicules, which are separate. All of them have spongin. And then class Demaspongiae has these silica spicules that are not shaped with 6 rays specifically, whereas the Hexactinellida, I'm going to get that down by the end of this, have ones that have at least 6 spikes on them, essentially. All right, so that's the end of our video. Make sure that you finished up your notes and be ready to do some more sponge stuff next class. See you then.